Okay, can you all hear me, everyone? Hello? Hello? Can you hear me? Hello?
Yes.
Okay, I'm going to start immediately. I will not wait. Now, I was thinking of revising the paper 6 document that I sent you, but I then thought that I'm going to solve with you an exam. I don't think you have access to this exam, November 2023. You don't have this exam, so we will solve it together. This is always the best practice before any exam is to solve past papers. So let's solve this one together. It's a typical exam that has everything that needs to do with paper 6. Okay, let's read the question together. Unfortunately, I cannot write here because it's password protected and I couldn't figure out how to remove the protection. So we'll read it together. They are here talking about pigeons. These are large birds found in Antarctica where the temperatures can be very low, and the fairy pigeons are smaller birds that live in the same area where the temperature is much warmer. And the body temperature of both species of pigeon is maintained at 38 degrees. A study investigated the rate of heat loss from a pigeon with a large body compared with a pigeon with a small body. Now, they did not get the pigeons, read along with me, because they used something instead of the pigeons. They used a large beaker, 250 centimeter cubed beaker to represent the larger pigeon, and a small test tube to represent the fairy pigeon. And the students used this method. Step number 1, they labeled the beaker A and the test tube B. Step 2, they drew a line on beaker A and test tube B 5 centimeters from the bottom. Step 3, they use hot water to fill beaker A up to the 5 centimeter mark, and they placed a thermometer in the water in beaker A. When the reading on the thermometer has stopped rising, they measured the temperature of the water. Record this as the starting temperature. Leave the thermometer in the water throughout the investigation, and then they started the stop clock. After 1 minute, measure and record the temperature of the water in beaker A. Measure and record the temperature of the water in beaker A every minute for a total of 5 minutes. Add hot water to test tube B to the 5 centimeter mark. Repeat steps 4 to 7 using test tube B instead of beaker A. And then they ask you for the table. Now, I just want to make sure that you understand the experiment, maybe because I was reading a little bit too quickly. So I cannot scribble on the exam, so I'm going to scribble here. What do? They want to know the effect of temperature on the loss of temperature, the effect of size of the pigeon on the loss of temperature. They have a large bird and a small bird, but they did not get the bird. Instead of getting the birds, they got a large beaker and a small test tube. And they filled the beaker and the test tube with the same volume of water, 5 centimeter water and 5 centimeter water. And they started with hot water. Okay, they started with hot water and then they measured the temperature of the water every minute. So what do you expect for the temperature of the water to happen every minute? The temperature of the water will, of course, decrease every minute. Okay, now what I'm asking you, what do you expect? The larger beaker, beaker A, or the smaller test tube B, which will lose heat faster, A or B?
A.
No, of course, B. A is big, it's a big bird, and it's storing heat inside its body. It will lose heat slowly, but B will lose heat faster. It's small. Like you have a big glass of milk and a small glass of milk, and they are both warm. Which one will become cool faster? Which one will lose heat faster? The big glass of milk or the small glass of milk? What do you think? Who said A?
The small.
Yes, the small. Okay. Here you're confused. It's not a rate of reaction. It's not a reaction. Here they are measuring the rate at which heat will be lost depending on the size. So the independent variable here, what did they change? What's the independent variable? Because they will ask you. Wake up with me. You have an exam tomorrow. What is the independent variable? They got a big bird and a small bird. They got a big beaker and a small beaker. So what did
The
they change?
different sizes.
Exactly. The independent variable here is the different sizes. And the dependent variable is the rate at which heat was lost, or the rate of heat lost, because they measured the decrease in temperature every minute. They started, for example, with a certain temperature, 100 degrees. Every minute, the temperature will drop. Let's say the 100 degrees will drop to 97, to 92, to 85. So the temperature is decreasing with time. And they stopped doing that after 5 minutes. And then they did the same for the smaller test tube. Let's say they started with temperature 100, they should start with the same starting temperature. And then they measured the drop in temperature every minute. Of course, the beaker B, it has like the heat will escape. It has less heat. It has less internal heat. So that more heat will be lost because it's small. So let's say after a minute, it will drop from 100 to 85. So the drop will be bigger. Okay, so let's look at the results. Here I jumped and I told you the results, but assumingly you will know the results from the table of results that is given here. The starting temperature for beaker A was 80 degrees Celsius, and for test tube B was 79 degrees Celsius. So can I stop you here and tell you, is this correct? The starting
source of
temperature
error.
Initial
is a source
temperature
of
must be
error.
the same.
Yes, the starting temperature should be the same, but it's not. Maybe it was difficult to get both temperatures to be the same. Now, beaker A, after 1 minute, it dropped from 80 to 69.5. Beaker 2, it dropped from 69.5 to 62 to 55.5. At minute 4, 51. At minute 5, they will tell you to read it from here. Now look at beaker B. The starting temperature was 79 degrees. After 1 minute, it dropped from 79 to 65. And then after 2 minutes, from 65 to 52, and then 47, and then 41. If you notice, the test tube B was losing heat faster. Every minute, it was losing more heat than beaker A. Now, let's not jump to conclusions and let's see what they want. Figure 1.2 shows the thermometers for beaker A and test tube B after 5 minutes. Of course, they will tell you, what is the temperature at beaker A? 3, 4, 5, 6, 7, 8. It's very easy. What's the temperature at beaker A?
48.
It is 40. Can't hear you.
48.
Yes, so up to now, it's very easy. And at test tube B, the temperature is 31, 32, 33, 34, 35.
35.
35, very easy. Of course, you will write the answer here or where they ask you to answer. Unfortunately, I cannot write. Now, this is important. This is 4 marks. Prepare a table and record the results shown in figure 1.1 and figure 1.2 to an appropriate number of decimal places. So you need to make a table in every paper 6 exam. This is a classic exam. You have to make a table. So you have here the information. You have the starting temperature and you have the temperature drop every minute. So how will you design your table? I already know how I will design mine. I'll give you a minute to think about it. If you have a paper and a pen, try to do it. What is the independent variable? This is the tricky table, by the way. What is the independent variable? What are you controlling? And what are you measuring?
The starting temperature.
The starting temperature is the independent variable. What are you like? What did you control? What did you decide? Didn't you decide that you will
The size of
1 minute
beaker and the
intervals?
test tube.
Excuse me?
I
Okay,
changed the sizes of the beaker and the test tube.
yes, the size of the beaker is the independent variable. Good. Okay, so this is one variable that you controlled, and you also controlled something else. Like, I want to ask you this question because I want to know which value will you put on the left-hand side of the table and which value will you put on the right-hand side of the table. Let's start it with the easy question. What are your results? What are you measuring at the end of each minute?
Temperature.
The temperature. Okay, I'll get a screen. So the temperature will be on the left-hand side or the right-hand side of the table. If my table is divided
Right-hand
into...
side.
Yes, okay, any table, at least 2 columns, okay? Put this in mind. So On the right-hand side of the table, you will have your dependent variable. In this case, it will be the temperature, and don't forget the units in degrees Celsius. Fine. And then one of you told me that the beakers that are of different sizes, they are the independent variable. Correct. So I can put the beaker here, beaker A and beaker B. Why? Because I changed the sizes of the beaker. They are my independent variable anyway. And what did I measure every minute? I measured the temperature. So what do you think I'm going to put here in this column?
Time.
Excellent. Time. Okay. So time, I think it was in minutes, every minute. Yes. So you have to put the units. So time 1, 2, 3, 4, 5. And for beaker B, time 1, time 2, times 3, times 4, times 5. And you will record the temperature at each time. You can design it this way. This is very acceptable. Or you can do it differently. You can put the time, minute, minutes, don't forget the unit. Time 1, time 2, time 3, time 4, time 5. Okay, 4. And the temperature for beaker A and beaker B. This is also very simple and very clear. So both are acceptable. Okay, so this is the time. You have 5 minutes and you will measure the temperature every minute for beaker A and beaker B. This will be an easier table because, and of course, don't forget the unit. This will be an easier table because you can easily compare the temperature drop at beaker A and at beaker B. So do you understand the table? Will you do it on your own? Like if we were not solving it together, can you now construct a table like this on an exam? Now I'll leave this answer for you. In order to prevent being confused on a table, always ask yourself, what is the dependent variable? And put the dependent variable on the right side of the table. This is at least going to guide you. This is going to be the first step. So I hope you can do something like this alone. Okay, let's go back to my exam. So we will put the results and you will put the result for time 5 and you will put it on the table. This is the table out of 4 marks. Now the rate of heat loss can be calculated using the equation. Rate of heat loss is the change in temperature over time. This is a very easy question. Using the results, calculate the rate of heat loss in beaker A and the rate of heat loss in beaker B or test tube B during the 5 minutes of the investigation. Include the units. Space for working. Okay, can you tell me how can you calculate the rate of heat loss for beaker A? Now 5, it was, at 5 minutes, it was 48. So the value here is 48. And minute 1, it was 69.5. How can you get the rate of heat loss? They gave you an equation. Change of temperature over time. What will you do?
Subtract.
Yes, you will subtract. And then subtract what from what and divide over what quickly?
69.5 minus 48 over 5.
No, you're wrong. 69.5 minus what?
80. 80.
Number 1, beaker A, the final temperature, or at 5 minutes, it was 48. Where did you get 80 from? So it's going to be 48 minus 69.5. The value is going to be a negative value because there's a decrease. Someone said 69.5 minus, yes, she's not wrong, because if you don't want to give a negative mark or a negative answer. So again, I'll leave it to you. What will you subtract? At 5
Dr.
minutes,
Manar, I have a
it's
question.
48, not 80 something. Yes.
هو مش المفروض نعمل subtract ال starting temperature اللي هو ال 80 minus ال 48؟
Starting temperature was 80 at 1 minute. Yes. Oh, this is from where you got the 80 from. I was not looking at that. I was looking at this. Oh, yes, you're right.
Yeah.
Okay, so tell me, you're right. I'm just in a hurry because I only have 40 minutes. Okay, what will you subtract? What's the final temperature?
80 minus ال 48
48
هتبقى
divided
32.
by? Okay, divided by?
32 divided by 5 هتبقى 6.4.
Okay, let me see the answer. You are correct. Where is it? Yes, excellent, 6.4. Okay, now the same, you will do the same thing here in the other test tube. 79 minus the temperature at 5 minutes, which is, we said 35 divided by 5. Okay? Any questions in this question? This is easy. Suggest the effect of the penguin body size on the rate of heat loss. Now, how will the body size affect the rate of heat loss?
The smaller the body size, the greater the heat loss.
Exactly, exactly. The smaller the body size, the faster the heat loss. Or you can write it in the opposite way or in the opposite direction. The bigger the body size, the slower the heat loss. Okay, now these questions, we already answered them. Identify the independent variable. What did we change in this investigation?
Different sizes.
Very good, different sizes. Identify one variable that should be kept constant in this investigation. Now that is
Temperature?
easy. The temperature.
Volume.
Okay. The temperature of what? No, try to avoid the
Starting
temperature.
point.
The starting. Okay, it's not wrong. What else can we keep constant? Let's think of something else.
The water volumes.
The water volume, correct. The water volume. Okay. Now, this is another experiment that is a very common one, by the way. We have cubes here of agar jelly. They are used as model cells. The agar jelly are colorless and they can be stained pink with an indicator. When placed in an acid solution, the acid will diffuse into the agar jelly cubes and the pink color starts to disappear. When the acid has reached the center of the agar jelly tube, the agar is completely colorless. This is shown in figure 1.3. So I hope you understand what they did here. They cut cubes of agar. They are of the same size. They did not change the size. And they started off with a pink color, and then they surrounded it, they soaked it in acidic solution, and they measured the time needed for the cube to change from pink to colorless. Okay? Now, I want to ask you a question. How did the acid go inside the jelly? How did it move?
Diffusion.
Diffusion.
By
Very good.
diffusion.
Okay, very good. Now here is the investigation question. Plan an investigation to determine the effect of temperature on the rate of diffusion in the model cells. 6 marks. The classic question that we have been solving from the beginning of the year. Okay, tell me. I'm going to get a screen while you're thinking. How can you change the temperature or study the effect of temperature on the rate of diffusion? Very easy and classic question. Come on. How do you write this question? What are the 6 points?
The independent variable.
Yes, yes, wake up with me. The independent variable. What will you change?
Temperature.
Very good. Change of temperature. And what are your results? What is the dependent variable?
Rate of diffusion.
Rate of diffusion, yes. How will you measure the rate of diffusion? What will you actually do?
Time for the.
Very good. You will measure the time needed for the cube to change from pink to colorless. Okay, good. So here we marked out our variables. Now, what are the steps? What will you do? And in the steps, you will tell me what are the constant factors. I don't know why you're slow. What are the steps? What will you do? What's the first step?
Put cubes of the same size and volume.
Excellent. First, you will cut agar cubes, okay? And you have to mention here that they are the same size, the same surface area, the same volume, whatever. And then what will you do after you cut cubes of the same size?
Thermostat controlled water baths at different temperatures.
Of course, you will soak them, okay, this is the keyword, excellent, thermostatically controlled water baths. And then what do you put on them? You will soak them in what? What will you put on them? In acid, yes. Of course, this acid is of the same what?
pH.
Exactly. Exactly. This acid should be of the same concentration, same pH, same volume. You have to use the same amount of acid. Very good. And then I think that's it. You will get beakers and you will put beakers of the same size, the agar cubes, and you will soak the agar cube with the equal volume of acid. Okay, these agar
دكتور
cubes
منال
they are
عندي
pink in
سؤال.
color. Yes.
هو can we change إن احنا نحطها في thermostatically controlled water bath بحاجة تانية؟ يعني مثلا oven.
No, because an oven is used with solids. Any beaker is
ما هو
put in
مش
a water
agar
bath.
jelly ده solid.
Yes, but if you put it
It's
in
a
an oven,
cube.
it will melt.
بس مش ال water ممكن affect the concentration of the acid.
No, the water is going to be acidic. You will soak it in acid. I did not say you will put it in water. You're confused.
ماشي. Okay.
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 So
Okay.
you will get the beaker. Okay. And this will have the agar. And the beaker will be inside a thermostatically controlled water bath. So the water is here. Inside the beaker, I don't know why
Okay.
it's disappearing. This is the acid. Okay, my dear? Okay.
Okay.
This
Okay.
is
Thank
water.
you.
And this is the acid. And because if you put the agar in the oven, the agar will melt. The agar melts very quickly. It's
Okay.
like wax. Okay, my dear? Good.
Okay. Thank you.
So we are in the steps. I think we made the steps clear. We will put equal sizes of agar in equal beakers. We will put each one in a thermostatically controlled water bath. And please give examples. Let's say cold. You don't have to give numbers. Cold and warm and very hot water baths. And then you will put an acid, equal volume and concentration of acid, and you will simply get a stopwatch and you will measure the time needed for the agar to change color entirely from pink to colorless. Is it clear? It's a very, very easy and classic investigation equation. And then the last two sentences that we write, the classic sentences, very good. We repeat at every temperature, we change the temperature, not the pH. Don't forget. We repeat at every temperature and then we calculate the average. And for safety, of course, we will be using acids. So we don't want the acid to touch our skin. We will wear gloves or we will make sure that the acid does not come next to a flame because it can be flammable. Okay. Now, I keep on telling you many times that in this question, you have to write it in points. Look at the mark scheme. I'll show you the mark scheme so you know for yourself that when you come to answer an investigation question, it is answered exactly like this because this has been the style of answering in the recent years. And starting 2019 onwards, investigation questions, if you noticed, that they answer it like this. Independent variable, at least two different temperatures. Use the word different, please. Detail of method, here are the steps. Method of maintaining the different temperatures. Here they mean the thermostatically controlled water bath. Method of cutting the agar cubes. It's better to use, even to be more detailed, I will use a knife to cut the agar cubes. Enough acid to cover the tubes or alternative wording. We already said that we will soak it in acid. Point 5 and 6 and 7. Variables kept constant. Maximum 3 from. Okay, so use the word same size of agar, same shape of agar, same type of agar, same concentration, same pH, same volume of acid, same type of acid, same type of indicator or name of indicator. So we mentioned 3 of those at least. We did say same size, same surface area, same volume, same volume of acid, same concentration of acid. So we will get the full mark. Dependent variable, we said it, the time taken for the cube to become colorless, or the idea after a set period of time, how much of the cube has become colorless. The same thing. Either you will measure the time needed for the cube to become colorless, or you will leave it for 5 minutes and you will measure how much of it became colorless. But the first one is easier. Okay, measuring the time for the cube to become colorless. And then look at 9 and 10. Reference to at least 2 further replications or repeat investigation at least 2 more times. So it's not only enough to say repeat, but say I will repeat at least twice and calculate average. 10, relevant safety precaution, anything safety. I'll wear goggles, I'll wear gloves, I will cut agar on a solid surface, cut away from your hands, alternative wording. Nice. So the investigation question, answer it like this. Independent variable, 2 dots, and write it. Details or steps, 2 dots. Dependent variable, 2 dots. Just exactly like the way we do it. Okay, you answered this question very well. The length of the side of a cube of agar jelly is 1 centimeter. Calculate the surface area to volume ratio. Surface area and volume is one of the equations that you have. How do you measure the surface area of a cube? I will not tell you. You will tell me. How do you measure surface area? Come on. Are you with me or you're gone? Hello? How do you measure surface area? You don't know how to measure the surface area?
A grid.
What do you mean grid? It's a cube. Oh my goodness. It's a cube. And it
6
is
times the S power 3.
What did you say?
Measure sides.
We want the surface
6
area and
times
the
S
volume.
power 2.
Yes.
Measure sides.
Huh? It's 1 times 1
6
because they
times
said
S power 6.
cubed. 1 times 1 is 1 times 6. So the surface area here is 6 centimeter cubed. Squared or cubed. This is surface area. 6 centimeter cubed. Now the volume. How do you measure the volume of a cube? Oh my God, you don't know
The
how to
side
measure the
length
volume?
power 3.
Yes,
that's
1 times 1
Width
times
times
1.
height times length.
Correct. And they said that it's all 1. So the answer will be 1 centimeter cubed. Now they want the ratio. How do I get the ratio of surface area to volume?
6 to 1.
6 to 1. Yes, you are correct. I don't know why you're not sure of your answer. You are correct. Okay, surface area and volume. Don't forget those. Where am I? So let's look at the answer. Yes, surface area 6 to 1. Very good. So the ratio is correct. So until now, we're doing a very good job. Now, the second question, we finished the first question. Paper 6 are 2 questions. Figure 2.1 is a photograph of a lizard. Definitely, they will ask you to measure CD. You will measure CD, you will use your ruler. It will be in centimeters, and you will convert that to what?
Millimeters.
Very good. And how do you do it?
Times 10.
Times 10. Okay, don't convert it to micrometers except when you are asked in the question. If you're not, you will keep it in millimeters. So line CD represents the length. Measure the length of the CD, whatever. Calculate, and they told you millimeters. Calculate the actual length of the lizard using the formula and your measurement. Very good. So they actually gave you a formula. What do they want? They want the actual length. So we always use our triangle. What's our triangle? Quickly. The triangle is what? I am. They definitely gave you the magnification. Let me see the magnification. 1 minute. Where is the magnification? Yes, it's times 0.6. So let's go back to my screen. So the magnification is times 0.6. And you want the actual. This is what they want from you. The size of the image. Let's say we measured it and it was 20 millimeters. So I will simply divide 20 by 0.6 and the actual will be X. I don't know what the answer is now. Also millimeters. If they ask you to convert this to micrometers, what do you do?
Times 1,000.
Very good. 1 millimeter is 1,000 micrometers. So I'll simply multiply X times 1,000. I hope you're with me. Maybe I'm moving quickly because Zoom is going to end soon. If we don't finish the exam, we'll go back. Okay, now, this is a figure 2.2. It's a photomicrograph of lizard blood cells. This is the red blood cell, and this is the nucleus. And this is a photomicrograph of a human blood cell, showing you a white blood cell here. Magnification times 900. Don't forget that magnification has no units. Magnification, its unit is times. State 2 ways the lizard blood cells shown in figure 2.2 are different from the human blood cells shown. Now, this is very, very easy and a complete waste of time. You want to compare those blood cells with those blood cells. Now, what will you write? This one, this is a blood cell, and this is the blood cell. This is the white blood cell. So you will compare this with this. See? There's a very obvious difference. Give me 2 differences. The red blood cell of the lizard, what does it have? Excuse
Figure
me?
2.2 have nucleus in the red blood cells, while figure 2.3 have no nucleus in the red blood cells.
Yes, of course. This is, of course, a very obvious difference. Here, there's a nucleus, and in our red blood cells, we don't have a nucleus. Okay, it's going to cut off. I'll go back in 10 minutes, fine? Now, another difference, quickly, please. So this is small. I just gave you an answer. So they want to
lizard oval.
oval, very good. And our red blood cell is more
Biconcave.
Circular.
circular. Okay, it is biconcave, you are correct, but it's not visible in the picture because this is the front view, not a side view. And this is smaller and this is larger and this is oval, this is rounder, this has a nucleus, this has no nucleus. Okay, I will not waste time here. Figure 2.2 shows one white blood cell. Draw a large diagram. Large diagram. How many marks? 4 marks. So look, this is a real exam here. And look at the space provided. They gave you this space. So please use the space. Like draw it this big. Okay? Don't use this space and then expect to take the full mark. No. You will draw the white blood cell. Now, the white blood cell, obviously, it is nearly circular, but the difficulty will be drawing the lobe nuclei. So you just need to do your best. You just need to do your best. Unfortunately, I cannot draw it here because it is password protected, but I'll take a picture with my phone and I'll try to draw it with you. Or let us go back to this question because Zoom is going to end because there's another important one here, and then I'll go back to the drawing. Okay. Hemoglobin is a protein found in human red blood cells. Hemoglobin carries oxygen. Athletes from a low altitude, height above sea level, location, train at high altitude in order to temporarily increase their hemoglobin levels. Scientists studied how long the increase lasted once the athletes returned to the low altitude location. I hope you understand the question. Some athletes, they go and they train on the top of a hill at high altitudes to increase their red blood cell count. Table 2.1 shows the results. This is the number of days after returning to low altitude. And this is the mean mass of hemoglobin per athlete. Identify the dependent variable. Even if you don't understand the experiment very well, what is the dependent variable? On which side of the table? Left or the right?
Right.
mass.
The right. Very good. So it's the mean mass of hemoglobin. Of course, they will tell you to plot a graph. Yes, exactly. Use the data in table 2.1 to plot a line graph on the grid to show the effect of returning to low altitude on the mean mass. So we will not waste time. This is 4 marks. What do I plot on the x-axis?
Independent
Dependent
variable.
variable.
Dependent variable, good, which is which? The left side of the graph or the right side of the graph? The number of days. So you don't even need to understand the investigation. Here they want you to plot the graph, so I will write before I start number of days after returning to low altitude. And here there are no units, it's just number of days. And on the y-axis will be the mean mass of hemoglobin per athlete. I'll just copy it exactly as it is in grams. And then what do we do? Unfortunately, I cannot here do any scribbling, but wait, I can. 1 minute. Let me annotate. Yes, look, good. So I'm going to start with what do we do with our graph? I'll put a 0 here, and then I divide it into equal boxes of what?
5.
5. Okay, I'll do it quickly. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, until you divide it here. And then you do the same here. Let's say these are boxes of 5. I'll do it randomly. I think I am... Okay, this is very random because we don't want to waste time. And then I'll go to my values. Here, the maximum value on the x-axis will be 40. So I want 40 to end here. I want 40 to end here, or maximum 45. On my y-axis, my maximum value is 650. So I want my 650 to end here. Okay, now I am going to send you this, like we need to do that. I don't know, how can we do it? Let us do the
ممكن
y-axis.
نعمل every boxes of 5 5.
No, I don't. What you mean, the x or the y-axis? The y-axis, I want
ال
650
X.
here.
لا
Okay,
ال
let me do
X.
okay. The problem is that I did not do it properly. I need to divide it into boxes of 5 properly. I'm in a hurry because Zoom is going to end. Okay, then I have to do it properly. So just be patient. Okay, 1. One, 2, 3, 4, 5. I make a mark. 1, 2, 3, 4. And please make a mark. Don't just write a number without a mark. 1, 2, 3, 4, 5. One, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, so now we did it properly. How many boxes? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 boxes. Good. It looks easy. let's go up. What's the maximum value we said? The maximum value on the x-axis is 40. So we want the 40 to be at the end of the axis. If we did 10, 20, 30, 40, 50, this is good. Or 10, if we did every 3 boxes, 10, 1, 2, 3, 10, 1, 2, 3, 20, 1, 2, 3, no, 30, no. So the easy way out is 10, 20, 30, 40. This will be acceptable. Anyone has any ideas?
Yes, you can do it by
Yes.
multiples of 4.
4?
It's
4,
10
I want
boxes for 4.
10 boxes with what?
For the last box will be 40.
Every box is how much? Tell me. Every box is the value of what?
4.
4. So this is
8,
4.
16.
Okay. And the 40 will be here. This will be perfect.
Yes.
Okay. Then fine. Then we will do it every box of 4. Let me look at the values. It will be easy to plot. 2, 4, 7. Yes, it will be very easy to plot. If every 5 boxes has a value of 4, so every small box will have a value of what? 4 divided by 5. Every small box will have a value of 0.8. If it's easy to plot for you, this is acceptable. If it's easy to plot for you, this will be fine. Now, the y-axis, what's the maximum value? 650. How many boxes do we have on the y-axis? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So how will you divide that? You tell me. I want the 650 to be here, up there. How can you divide that? 120, 400, 300, 400, 500, 600, no.
I guess each 5 was 55.
Wait a minute. Let me. Problem is that I like it multiples of 5. Okay, 650 divided by, we said how many boxes? 1, 2, 3, 4, 5, 6, 7,
5
8, 9,
boxes.
10, 11, 12. So 60 divided by 12. No, this is not nice. So let's say 100 divided by 12. Okay, what did you do? This is not an easy graph. This is good practice. So what did you tell me? We said every box will be how much?
55.
55. And then it's going to end with 650. 55. 655 plus 55. This is 110. Then plus 55. This is 165. I don't like these values, but if they meet the criteria, then it's okay. 220 plus 55, 275. You know what we're doing? I'm adding 55 every box plus 55. That's 330. How will it end? 385. 6 what? 650? So if the boxes each is 55, it will end at what? I can't hear you.
660.
Okay,
660.
660. Very good. Okay, so the 650 will be up there. Good. Okay, the values, I don't like them very much, but they meet the criteria. They are equally distributed. You don't have definite values, by the way. You don't have definite values. You only need to meet the criteria where that you have to label your axes with the units and you have to use more than half the grid. I'll show you the mark scheme because the mark scheme says clearly that you have to follow those criteria when you come to do any graph. Let me erase everything I have here.
Doctor,
we can
if I
see.
have mine, but it doesn't end at 650, but it's more than half the graph, صح?
No, it can be, it's okay. It just needs to be more than half the graph. Okay, but a more with a
Okay.
little bit, not a more by one block. Okay, like a more by a decent number. So here, where are we? Axes labeled with units. I keep on telling you the units. If you don't put the unit, you lose a number. You lose a mark, i.e. number of days after returning to the low altitude, and mean mass of hemoglobin per athlete in grams. So the